This video is about antigen recognition by immune cells, adaptive immunity and antibody, and this study note is from EPG Pathshala, a government site, Indian government, and I would like to express my sincere gratitude to the development team for developing this study note and using this for study purpose. Thank you. Next topic, antigen recognition by immune cells, adaptive immunity, antibody. Antigen recognition by immune cells, adaptive immunity and antibody. Immunity, adaptive, antibody, antigen, epitop we will be learning. Light chain, heavy chain, B lymphocyte, T lymphocyte, humoral immunity, isotypes and effector function. And then adaptive immunity and introduction, antibody, unique features of tool of humoral immunity, antibody, important discoveries or research work related to antibody. Then important discoveries or research work related to antibody along with structure of antibody, light chain, heavy chain, J chain, hinge region, disulfide bond, carbohydrate group. Experiment of A. Teselius and E. A. Cabot. Experiment of Rodney Porter and G. M. Edelman. Structure of antibody, light chains, heavy chains, J chain, hinge region, carbohydrate group, sulfide group, etc. Then types of antibody, IgA, IgM, IgA, IgG, IgM, IgA, IgE, IgD. Functions of antibody, opsonization and complement activation. Along with antibody dependent cell mediated cytotoxicity or ADCC. Then transcytosis, as along with that passive immunity. And later we'll be having a quick summary about that. The study of the chapter will help in understanding the following: what is adaptive immunity and such kind of things. So let's have an introduction. The chapter focuses on the biology of antibody, which is an important effector molecule of the humoral adaptive immunity. Antibodies or immunoglobulins, which were earlier called magic bullets, are released in millions on activation of B lymphocytes. The study of structural detail of the molecule is important to understand its function, which is of great help to customize and manipulate it commercially for available diagnostic and therapeutic uses. The important attributes of specificity and diversity gives it the convenience to act at the strategic locations, targeting variety of antigens entering through different routes. The antibody is not only released in the body fluids, but is also located on the B cells as membrane receptors, where it is a part of antigen recognition unit of B cell. The magic of the antibody is clarified once we analyze its role as a static molecule on the B cell, IgG or MIG, and the mobile effector molecule in blood and other secretions. Adaptive immunity. The adaptive immunity is given by the activation of two types of cells in the blood, that is B and T lymphocytes. These specialized cells are named on the basis of their immunological training in the bone marrow or thymus respectively. Adaptive immunity is also called acquired immunity, as it starts after the exogenous or endogenous antigen enters and infects the body. B cell attacks the exogenous outside antigen and T cell is deputed for endogenous inside antigen. B cell attacks the exogenous or outside antigens and T cell is deputed for endogenous or inside antigen. It starts a few days after infection, as it gives the first chance to the immune mechanism to provide the first line of defense. Adaptive immunity has three important roles of recognizing, eliminating and remembering the pathogen by producing few memory cells. It also has the power of giving immunity specific to the antigen due to presence of different receptors, that is RCR and TCR, located on the B and T cells. It is called acquired immunity as it is a specialized defense mechanism which adapts on acquiring infection. The existence of two different types of lymphocytes, that is B and T lymphocytes, was first reported by J.A. Perry Miller and G.F. Michael in 19 66. Both the lymphocytes work efficiently in a cooperative manner to give humoral and cell-mediated immunity. The response given by B cells is called humoral, and the response given by T lymphocytes is called cell-mediated. T4 cell-mediated, and C is dealing with endogenous antigens. So endogenous in the sense cell-mediated. Remember like that. Then these two subpopulations of T cells with CD4 and CD8 on their membranes called T helper or TH and T cytotoxic cells or TC, where TH cells decides what it is to be activated, that is cell-mediated immunity or humoral immunity. There is another subpopulation of T cells called the T suppressor cells, TS. They are the negative regulators of immunity and suppress the immune response if there is overreaction in the body or reaction for self-proteins. They are thus called immunoregulator cells. TH, TH and TC. T helper and cytotoxic cells, TC or TH. TS will be there. Then the mature B or T cells which waits for encounter with the antigen for the first time is called naive lymphocyte. Adaptive immunity differs from innate immunity in its antigenic specificity and presence of memory cells. The memory cell membrane has the same receptors as present on the activated lymphocyte, but recognizes the antigen faster if it dares to enter the body again. BCR, B lymphocyte, mature in bone marrow and kills exogenous antigen, matures in thymus and kills endogenous antigens. TCR, BCR. Naive B cell and memory B cell. Naive B cell, virus A2, no contact. Then virus A1 contacted with this memory B cell, specific for virus A. Antivirus A antibodies are produced. Antivirus A antibodies are produced. Then antibody. All the vertebrates are gifted with the ability to form different sized multifunctional glycoprotein called antibody for giving the protection against the invading antigens. It is released in the blood and other body fluids like milk, saliva, tears, etc. Because of which this type of immune response is called a humoral immunity. Humor means fluid. The term antibody has originated from the German word Antikörper. It was first coined by P. Ehrlich in end of 19th century and was explained in his selective theory as side chain receptors. E. V. Behring and S. Kitasato described it as an agent in blood that was capable of passively transferring the immunity. They suggested that there exists an entity which could act against the anti-offending toxin. They discovered diphtheria antitoxin serum, which was the first immunological approach for tackling the infectious diseases. The antibodies may be located on the membranes of the B cell or they may be secreted. The membrane-bound antibody, MIG, membrane-bound IG, acts as a receptor on the B cell, BCR, for AG recognition, and the second form is for neutralization, clumping, precipitation and agglutination, etc. of AG. The antibody present on the B cell makes it antigen specific by recognizing epitopes present on the antigen and activating the same cell after the signal is transduced. The B cell is also an antigen presenting cell where it first endocytose the antigen and display it with the membrane molecule MHC2. It is then activated by cytokines of TH cells which have CD4 marker on their membranes. The antibodies may be located on the membranes of B cells or they may be secreted. The membrane-bound antibody MIG acts as a receptor on the B cells BCR for AG recognition and are secreted from is for neutralization, clumping, precipitation, and agglutination, etc. of antigen. The antibody present on the B cell makes it antigen specific by recognizing epitopes present on the antigen and activating the same cell after the signal is transduced. The B cell is also an antigen-presenting cell where it first endocytoses the antigen and display it with the membrane molecule MHC2. It is then activated by cytokines of Th cells which have CD4 marker on their membranes. The B cell, once activated, proliferates and produces clones of antigen-specific B cells which finally change into the antibody-secreting plasma cells. The antibodies try to inactivate, neutralize, agglutinate, precipitate, and clump the microbes so that they may not multiply and colonize in the organs they invade. You could see a picture. Antigens, different sort of antigens. Antigen binding site. Antibody showing antigen specificity. A B cell is triggered when it encounters its matching antigen. The B cell engulfs the antigen and digests it. Then it displays antigen fragments bound to its unique MHC molecules. This combination of antigen and MHC attracts the help of a mature matching T cell. Cytokines secreted by the T cell help the B cell to multiply and mature into antibody-producing plasma cells. Are released into the body. Antibodies lock onto matching antigens. The antigen-antibody complexes are then cleared by the complement cascade or by the liver and spleen. Activated B cell. And unique features of tool of humoral immunity. Antibody. Antibody is an immunoglobulin. Antibody is an adapter molecule as it is a bridge between antigen and effector molecule. Antibody is a glycoprotein. Antibody is a heterodimer. Antibody is an effector molecule. Antibody is specific to a single epitope. Antibody is in the serum is heterogeneous. Antibody can act as antigen sometimes. Antibody raised against antibody is called anti-antibody. Antibody is of 5 types varying in the effector functions. Antibody can be monomeric or polymeric. Antibody is monoclonal and polyclonal. Antibody is a flexible molecule. Antibody is of 2 types, membrane bound and secretory. Membrane antibody is for antigen recognition as BCR, B cell receptor. Important discoveries or research in this area of antibodies. T-rich in 19. Antibodies are magic bullets to target human disease and gave selection theory of antigen and antibody. E. von Behring. Serum antitoxins got Nobel Prize in 1901. R. Tiselius and Elvin Cabat. Antibodies are serum proteins or immunoglobulins discovered in 1939. Karl Landsteiner in 1940 demonstrated that antibodies are specific in their recognition by using hapten. H. N. Eisen and R. Krush in 1940 discovered that antibody is divalent. PK reaction to study allergy. Biological function of IgD, biological function of IgA, gene arrangement in antibody production. Monoclonal antibody identified the hypervariable region on the antibody. Heavy chains are involved in effector function of antibody. Chemical structure of antibody. Then there are pockets in antibodies where antigen binds and interaction between the two is non-covalent. Antibody is divalent. Antibodies are specific in their recognition by using hapten, serum proteins or immunoglobulins, serum antitoxins. Landmark experiments to study the anatomy of antibody. Experiment of A. Tiselius and K. A. Cabot. A. Tiselius and E. A. Cabot in 1939 worked on immunized rabbits by using the technique of electrophoresis. By this technique, the proteins on the gel, when subjected to the electric current, are separated according to their size. They immunized the rabbit with the antigen ovalbumin. Then ovalbumin, OVA, a strong protein of fowl egg, is a good immunogen for the mammals due to its large size and easy availability for immunization. They took the serum of the immunized rabbit and divided it into two parts before electrophoresis. After the two parts which were electrophorized, one part was mixed with ovalbumin before electrophoresis. The second part of serum, which was not mixed with ovalbumin, showed 4 peaks corresponding to albumin and alpha, beta, gamma globulin. But in the serum mixed with ovalbumin, the gamma peak in the graph was absent. They concluded that the antibodies are in the gamma globulin fraction and hence named gamma globulin or immunoglobulins. With the advancement in electrophoretic technology, it is known that IgG, the major antibody, exists in gamma fraction, but alpha and beta globulins also contain other classes of antibody. Experiment of Rodney Porter and G.M. Edelman. The insight into the structure of antibody was given by Rodney Porter and Gerald M. Edelman, for which they shared the Nobel Prize in Physiology in 1972, though they worked independently on the molecular structure of the antibody. A large protein immunoglobulin was broken using different enzymes like pepsin and papain and reducing agents like mercaptoethanol. The results were then compiled to deduce the Y-shaped structure of antibody. Pepsin cleaved the immunoglobulin on site at site of aromatic amino acids like tyrosine, tryptophan, and phenylalanine, resulting in 2 Fab and 1 Fc parts. But the cleavage site of papain, enzyme obtained from papaya latex, is at the cysteine residues. The cleavage product of papain was a single piece with 2 Fab components. The Fc fragment was digested by the enzyme completely. Mercaptoethanol broke the secondary and tertiary structure of the protein by reducing the disulfide bonds, thus releasing the polypeptide chains of 2 different sizes called as light and heavy chains. The large heavy chain was of 55 kilodalton, but the light chain was of 22 kilodalton. A variety of antibody types was listed by deciphering the amino acid sequence. The amino acid sequence of the antibody was studied using cancerous plasma cells called myeloma cells. Variation in the amino acid sequence of that light chain was later obtained by Bence-Jones BJ proteins. Bence-Jones, I'm not sure of the pronunciation. Bence-Jones proteins are immunoglobulin light chains produced by the neoplastic plasma cells. They clumped when heated on high temperature. Based on the studies of BJ proteins, there are 2 types of light chains called kappa and lambda, differing in the constitution of constant region. The heavy chain and the light chain both have variable and constant regions. The constant region of heavy chain are of 5 types, determining the 5 classes of antibodies as IgM, IgG, IgD, IgE, and IgA. Gerald M. Edelman and Rodney R. Porter. Only look at this picture. This is the effect of proteolytic enzymes and mercaptoethanol on antibody. Disulfide bridge is there, heavy chain is there, light chain is there. Then pepsin cleavage will be taking place. So FAB12-TJ, papain cleavage, Fab protein. mercaptoethanol reduction, Fc fragments, HS light chains. Structure of antibody: light chains, heavy chains, J chain, hinge region, carbohydrate group, disulfide bond. This is a very important topic. Light chain and heavy chain. The antibodies are proteins made of 4 polypeptide chains. Do you know that? I hope. If you don't, listen carefully. The antibodies are proteins. Antibodies are The antibodies are proteins made of 4 polypeptide chains. The antibodies are proteins made of 4 polypeptide chains. The two large sized of 5 kilodalton each are called heavy chains, and the small sized polypeptide of 25 kilodalton are called light chains. The two different light and heavy chain organize to form a Y-shaped 3-dimensional structure of the antibody due to intra and inter disulfide bonds and other non-covalent interactions. The protein nature of antibody also contributes to the two functionally different N and C terminals with specific roles. N terminal shows variability in amino acid sequence compared to C terminal of polypeptide. The region of the heavy and light chain showing amino acid variation in each molecule is denoted as the region of the heavy and light chain showing amino acid variation in each molecule is denoted as VL and VH. VL in the sense variable region in the light chain, variable region in the heavy chain, and constant region as CH and CL. Region of heavy and light chain showing amino acid variation. So this is a structure, antibody structure. Variable portion, you could see disulfide bond is also there. Heavy chain, constant portion, light chain. Variable portion, constant portion, disulfide bond, heavy chain, light chain. Okay. The intra-chain disulfide bonds at a regular interval of 110 amino acids result in converting the peptide chain into a compact globular domain. Each light chain consists of a single constant domain called the Cl, whereas the heavy chain has 2 or 3 constant domains called CH1, CH1 and CH3. CH1, CH2 and CH3. Antibody domain structure: variable heavy, variable light, CH1, CH2, CH3. Cl, hinge region. Domains making heavy and light chain: yellow and green are the light chains, and blue and red are the heavy chains. The study of secondary and tertiary structure of antibodies show all the immunoglobulin domains have beta-pleated sheets, each consisting of 3 or 4 strands of antiparallel polypeptides connected by loops of variable length. The study of secondary and tertiary structure of AB show all the immunoglobulin domains have a beta-pleated sheets, each consisting of 3 or 4 strands of antiparallel polypeptide connected by loops of variable length. The two beta-pleated sheets are connected by the conserved disulfide bonds. The characteristic protein motif found in immunoglobulins is called the immunoglobulin fold. Protein motif, immunoglobulin fold. N-terminal variation in composition of heavy and light chain facilitate the execution of humoral response to a variety of antigens. Within the variable domains of light and heavy chains, there are short amino acid sequences with exceptional variability at 3 locations called hypervariable regions. They are also called complementarity determining region or CDR, as they are the regions which protrude out to form the peptide loops, giving a shape complementary to the shape of an antigenic determinant or epitope. There are three CDRs named CDR1, CDR2 and CDR3 which cluster to give the shape to the antigen binding site of antibody, also termed as paratop. AB-AG interaction occurs at epitope of the antigen and paratop site of antibody. The rest of the variable region is called the framework region, as it provides a scaffold to hold the hypervariable regions in place. There are two types of light chain in the monomer of antibody called lambda and kappa. The names lambda and kappa represent the Greek letter in Greek of the two scientists L. Kongold and R. Lipri, who first discovered them. Combination of both types of light chains is present in each individual, but the percentage composition vary in different species. There are 5 different types of heavy chains named as alpha, mu, gamma, delta, epsilon based on the difference in the constant region of the heavy chain. The antibody with 5 different types of heavy chains exists in an individual and is thus further called classes or idiotypes. Each isotype has not only a distinct physical and biological property but differs in its effector functions. The antibody with alpha chain is called antibody A, with mu chain is called antibody M, and accordingly antibody G, D and E. The subisotypes of the heavy chains also exist due to the small amino acid variation in the isotype alpha 1 and alpha 2. The subisotypes of antibody A and gamma 1, gamma 2 and gamma 3 and gamma 4 are the subisotypes of antibody G. The different isotypes and its subtypes vary in performing effector functions like opsonization, complement activation, neonatal immunity, etc. Most of the effector functions are mediated by the C-terminal of the heavy chain of the antibody as it binds with the receptors FCR on the cells like NK, mast cell, phagocytes, etc. The antibody is also classified as monomeric and polymeric. Monomeric antibody has 2 binding sites for an antigen, but the multimeric antibody has more than 2 binding sites. Monomeric and polymeric. Monomeric antibody has 2 folding sites for an antigen, but the multimeric antibody has more than 2 binding sites. This interaction of antibody with the antigen is possible because of flexibility in the 2 arms of the antibody by the presence of hinge region, especially in the IgG, IgD and IgE. The antibody which is specific for a single epitope is called a monoclonal antibody. The human serum contains a cocktail of antibodies specific for various antigens and it is called a polyclonal serum. The monoclonal antibodies are prepared in vitro by hybridoma technology. They are used as therapeutic and diagnostic agents. The hinge region. The hinge region is located between CH1 and CH2 domains of the antibody. If you want to see the figure, just travel a bit backwards. Yeah, here you could see. We're learning about hinge region. The hinge region is located between CH1 and CH2 domains of the antibody. It is mainly made of 2 types of amino acids, that is proline and cysteine, which are vulnerable to the attack by the proteolytic enzyme like papain. The proline amino acid provide the flexibility to the arms of AB. The hinge region is a region located between CH1 and CH2 domains of the antibody. It is mainly made of 2 types of amino acids, that is proline and cysteine, which are vulnerable to the attack by the proteolytic enzyme like papain. The proline amino acid provide the flexibility to the arms of AB, but the cysteine amino acid helps in forming the interchain disulfide bonds. The hinge allows the 2 antigen binding Fab region of each antibody molecule to operate independently so that it can bind the antigen epitopes which are distinctly separated. Though the arms are flexible, but its flexibility also hinders in saturating all the binding sites of the multimeric antibody. IgG3 has the longest hinge region amongst the subtype of IgG. The hinge region varies in the length from 10 to 60 amino acids approximately. Then J chain. These are small proteins coded by different genes which connect the units of the polymeric immunoglobulin. It is joining chain with a molecular weight of approximately 15 kilodalton and is linked by the disulfide bond to the C terminal of the heavy chains. Then disulfide bond. Heterodimeric antibody has intra and interchain disulfide bonds between the peptide chains, which gives stability to its three-dimensional Y-shaped molecular structure. Disulfide bond. And carbohydrate group. The immunoglobulin are glycosylated, which means the carbohydrate moiety is attached to it, which is important for the passage of the antibody through the biological membranes and placental barriers. All the isotypes have a different half-life. The shortest half-life of IgE, that is of 2 days, IgG of 3 days, IgM of 4 days, and IgG has longest half-life of 21 to 28 days. Shortest half-life is of IgE, that is of 2 days, IgA of 3 days. So we just mentioned about carbohydrate group. The immunoglobulins are glycosylated, which means the carbohydrate moiety is attached to it, which is important for the passage of the antibody through the biological membranes and placental barriers. All the isotypes have a different half-life. The shortest half-life is of IgE, that is of 2 days, IgA of 3 days, IgM 4 days, and IgG has longest half-life of 21 to 28 days. EAMG. EAMG. The long half-life of IgE is attributed to the FcRn, neonatal Fc receptor, which protects it from lysosomal degradation for a long time. So if IgE has shortest half-life of 2 days, then comes IgA, IgM, then IgG. Okay. Structure of antibody, a detailed structure. And you look at this figure, you could see portions like amino terminal, heavy chain, light chain, hinge region, CHO, carboxyl terminal, disulfide bonds, carboxyl terminal, disulfide bonds, effector function, then antigen antibody sites. Types of antibody. The 5 different types of antibodies, IgG, IgM, IgA, IgE, and IgD, perform different biological functions, and each type is important so as to kill or inactivate the pathogen entering through various routes in the body. First is about IgG. The IgG is a monomeric antibody having the highest concentration in the serum. IgG is a monomeric antibody having the highest concentration in the serum. It constitutes about 80% of the total serum concentration. It is a good complement activator, can cross placenta and gives the passive immunity to the fetus. Once again, the IgG is a monomeric subunit antibody having the highest concentration in the serum. It constitutes about 80% of the total serum concentration. It is a good complement activator, can cross plasma and gives the passive immunity to the fetus. It also mediates the opsonization process by the macrophages and the neutrophils. Based on the minor amino acid differences in the heavy chain, it has been divided into subclass, namely IgG1, IgG2, IgG3, and IgG4. There is a difference in the number of disulfide bonds and the size of the hinge region in the subclass of the isotype IgG. Out of the 4 subtypes, IgG1 is best in giving passive immunity to the fetus and in mediating the process of opsonization. So IgG3 is the best activator for complement G3. Its concentration is less in primary immune response but increase in the secondary immune response. So IgG is very important monomeric antibody. Monomeric, remember it's monomeric, having high concentration in the serum. It constitutes about 80% of the total serum concentration. It is a good complement activator. It cross placenta and gives the passive immunity to the fetus. It also mediates the opsonization process by the macrophages and neutrophils. And out of the 4 subtypes, IgG1 is best in giving passive immunity to the fetus and in mediating the process of opsonization. IgG3 is the best activator of complement. Its concentration is less in the primary immune response but increase in the secondary immune response. Then IgM. So we learned about IgG, then about IgM. It is the antibody present on both the cell membrane and in secretions. The membrane-bound antibody is monomeric, but the secretory form is pentameric. Then, though it can bind with 10 epitopes, but there is steric hindrance. IgM is the antibody present on both cell membrane and in secretions. The membrane-bound antibody is monomeric, but the secretory form is pentameric. Though it can bind with 10 epitopes, but there is steric hindrance as a result. It can interact with the maximum of 6 or 7 epitopes. It is existing in the serum with only 5 to 10% of the total concentration, and a very less amount exists in the mucosal secretions. It is the antibody for the primary response as it appears first in response to the infection. The pentameric secretory form of IgM helps to attack the pathogen quickly. It cannot cross the placental barrier due to its polymeric nature. It is the first antibody to be produced by the neonate IgM. The 5 monomers of the antibody are linked to each other by the joining chain or J chain. The J chain is synthesized in the plasma cell. The J chains also help in transport of the antibody across the epithelial layers as mucosal secretion. It is a best complement activator and forms big aggregates of agglutination. IgM. It is important to know that the IgM also include isohemagglutinin, which are the naturally occurring antibodies against the red blood cell antigens and thus determine the ABO blood group system. These antibodies might have originated as a result of some bacteria entering through the various routes as discussed earlier. These bacteria had antigenic determinant similar to the oligosaccharide antigen of the ABO blood group. This without the immunization of individuals, anti-A or anti-B or both may be present in the blood. It is important to know that IgM also include isohemagglutinin, which are the naturally occurring antibodies against the red blood cell antigens and thus determining the ABO blood group system. These antibodies might have originated as a result of some bacteria entering through the various routes as discussed earlier. These bacteria had antigenic determinant similar to the oligosaccharide antigen of the ABO blood group. Thus, without the immunization of individuals, anti-A or anti-B or both may be present in the blood. The pentameric nature of IgM does not allow it to cross the placenta, so incompatibility of ABO is not dangerous to mother and fetus. Here you could see group A, red blood cell type A. Antibodies in plasma, anti-B. Antigens in red blood cell, A antigen. Then group B, red blood cell type B. Then anti-A will be the antibody in plasma. Antigens in red blood cell, B antigen. Then group AB, no antibody but A and B antigens in red blood cell. Then group O, red blood cell type O, antibodies in plasma, anti-A and anti-B, but antigens in red blood cell, none. And IgA, it is the type of antibody which exists in the serum and in the mucosal secretions of the respiratory, gastrointestinal and urinogenital tract. This is the type of antibody which exists in the serum and in the mucosal secretions of the respiratory, gastrointestinal and urinogenital tracts. IgA is a type of antibody which exists in the serum and in the mucosal secretions of the respiratory, gastrointestinal and urinogenital tract. यहाँ क्या है IgA. So it is dimeric. ये dimeric है, trimeric और tetrameric वो भी हो सकता है in its secretory form. It is mostly transported as dimeric form. It is also assisted by the J chain for the polymerization and its transport to the mucosal surface. It is important as it coats the antigen and prevents its attachment to the mucosal layers. So ये antigen को coat करते हुए दिखाई देगा and that will definitely gonna prevent its attachment to the mucosal layers. So mucosal layers से attach होने से ये बचा के रखेगा antigen को. Okay. So thus, for protection against local infections, routes of immunizations causing local production of IgA is better than those producing antibody in serum. Once again, I'm going to read this. It is the type of antibody which exists in the serum and in the mucosal secretions of the respiratory, gastrointestinal and urinogenital tract. It is dimeric, trimeric or tetrameric in its secretory form. It is mostly transported as dimeric form. It is also assisted by the J chain for the polymerization and its transport to the mucosal surface. It is important as it coats the antigen and prevents its attachment to the mucosal layers. Thus, for protection against local infections, routes of immunization causing local production of IgA is better than those producing antibody in serum. As the complement receptors are absent on IgA molecule, so it is not a complement activator. It is a type of antibody which exists in the serum and in the mucosal secretions of the respiratory, gastrointestinal and urinary tract. It is dimeric, trimeric or tetrameric in its secretory form. It is mostly transported as dimeric form. It is also assisted by the J chain for the polymerization and its transport to the mucosal surface. It is important as it coats the antigen and prevents its attachment to the mucosal layers. This, for protection against local infections, routes of immunizations causing local production of IgA is better than those producing antibody in serum. As the complement receptors are absent on IgA molecule, it is not a complement activator. Then IgE, it is present in very less concentration in the serum, whereas biologically very potent to induce hypersensitive reactions against an allergen. So you might have already learned about hypersensitivity, 1, 2, 3, 4 types. I have a video on that. So let me think of that video. You could recollect that hypersensitive reactions and allergic reactions are to a greater extent related with IgE type 1 in particular. It is named antibody E because of antigen E present on the ragweed pollen. The presence of this antibody was first detected by O. Prusnes and S. Kussner in 1921, and the reaction is also called a PK reaction, Ilan-Flare reaction or whatever it may be. It binds to the Fc receptor present on the membranes of basophils and mast cells. The cross-linking of the allergen with the antibody bound to Fc receptors on the same cell results in giving the signal for the degranulation of the pharmacologically active reagents which cause a reaction like Whelan-Flare or hives, etc. IgD. It is present as membrane-bound antibody on the mature B cells. It acts as a receptor on B cells, so-called BCR. IgD, it is present as membrane-bound antibody on the mature B cells. It acts as a receptor on B cells, so-called BCR, IgD. Its biological function is not very clear. It is less than 5% of total serum immunoglobulin. So I guess IgD is, always open for research. We must research in this topic to find its biological function. IgD. BCR. Type of antibody and their properties. IgM pentamer, IgG monomer, secondary IgA dimer, secretory component, IgE monomer, IgD monomer. He chains mu, gamma, alpha, epsilon, delta. Number of antigen binding sites 10, 2, 4, 2, 2. That can also be stated as a valency of that one. So molecular weight, here you could see percentage of total antibody in serum, crosses placenta, IgM, no crossing, then IgG, yes, and other three never cross, IgA, E and D. The only thing which crosses is IgG. Yeah, IgG. Fixes complement, first two, that means IgM and D fixes complement, but other three A, E and D won't be doing that. Phagocytes, wait a minute, Fc binds to phagocytes in case of G. Mast cells and basophils in case of E. Yeah. Function IgM. Yeah, first one is IgM. So IgM functions, main antibody of primary response, best at fixing complement. The monomer form of IgM serve as the B cell receptor. The last IgD is a B cell receptor. Then IgG, main blood antibody of secondary responses, neutralizes toxins and opsonization. Main blood antibody of secondary responses. Main blood antibody of secondary response, neutralizes toxins and opsonization. Then IgA, I guess. Yeah, IgA-ED. Secreted into mucus, tears, saliva and colostrum. Antibody of allergy and anti-parasitic activity, B cell receptor. Light chain, antigen binding site, disulfide bonds, carbohydrate, IgG. Structure of IgG. Light chain, antigen binding site, disulfide bonds, carbohydrate, IgG, Y heavy chain. Then IgM is a pentamer. IgM and IgD, new heavy chain. IgM, CHO group, joining chain, new heavy chain in case of IgD. Then IgA monomer, secondary IgA dimer, alpha heavy chain and joining chain. Heavy chain, IgE, IgD, alpha heavy chain. Then it can also act as a pentamer, which one? IgD. Types of antibody, secretory and membrane bound. Membrane antibody, plasma membrane. Functions of antibody, the two different components of antibody, that is the Fab and FC, are for epitope-paratope interaction and effector function respectively. The various functions of antibody are opsonization. Opsonization, it is the coating of the antibody on the surface of the antigen due to the epitope-paratope interaction. The two different components of antibody, that is Fab and FC region of epitope. Opsonization, it is the coating of antibody on the surface of antigen due to epitope-paratope interaction, which makes the antigen more vulnerable to attack by both the phagocytic and non-phagocytic cells. The phagocytic cells like macrophages and neutrophils and some non-phagocytic cells have the receptors FcR for binding with the Fc of antibody. This interaction results in initiating the signal transduction pathway for the different mechanisms of phagocytic activity for the secured killing of the pathogen. So you could see opsonization, macrophage is there. Receptor antibody, antigen, bacteria. Opsonization, antibody, microbe, Fc, receptor. Then complement activation. Wait a minute, what is opsonization? I have some doubts. It is the coating of antibody on the surface of antigen due to epitope-paratope interaction, which makes the antigen more vulnerable to attack by both the phagocytic and non-phagocytic cells. And the phagocytic cells like macrophages and neutrophils and some non-phagocytic cells have their receptors for binding with Fc of antibody. This interaction results in initiating the signal transduction pathway for the different mechanisms of phagocytic activity for the secured killing of the pathogen. Complement activation. Complements are the sets of serum proteins which, when activated, make perforations in membrane of pathogen and finally kill it. Antigen antibody C1 complex, C2a and C4 C4b fragments, classical pathway C3 convertase, alternative pathway C3 hydrolysis, C3b and C3a fragments, C3b cleave C5 into C5a and C5b, cell swells and bursts, C5b, C6, C7, C8 and C9 together form the membrane attack complex. Complement activation. Then antibody dependent cell mediated cytotoxicity. This lecture is going to end, so I'm gonna quickly read things. Okay, the natural killer cells NK are activated for its cytotoxic function by antibody antigen interaction, and these cells have the Fc receptors on their membranes. Okay, antibodies bind antigens on the surface of target cells. Antibodies bind antigens on the surface of target cells. NK cells CD16 Fc receptors recognize cell-bound antibodies. Cross linkage of CD16 triggers degranulation into a lytic synapse. Tumor cells die by apoptosis. That is ADCC once again. Antibodies bind antigens on the surface of target cells. NK cells CD16 Fc receptors recognize cell-bound antibodies. Cross-linking of CD16 triggers degranulation into a lytic synapse. Tumor cells die by apoptosis. Then, as I mentioned, I'm gonna quickly read these things. Okay, transcytosis. The Fc component of AB assists in transfer of the antibody from the epithelial layer to the mucosal surface of the gastrointestinal, respiratory, and the urinogenital tracts. The Fc component of AB assists in transfer of the antibody from the epithelial layer to the mucosal surface of the gastrointestinal, respiratory, and the urinogenital tracts. It binds with the poly-Ig receptor on the basolateral side of the epithelial cell, which is endocytosed and then excreted in the mucosal surface. That is transcytosis. And passive immunity. The growing embryo in the mother's body, that is fetus, is protected by passive immunity by IgG, is selectively transported across the placenta mediated by neonatal Fc receptors, FcRn. Maternal immunogen is also transferred to the offspring through the transport of IgG across the gastrointestinal tract. Ag and AB interactions involve 4 types of non-covalent interactions, making it a reversible reaction. Antibodies can also become potent immunogen and can induce immune response. Based on the difference in light and heavy chains, they are classified as isotypes, allotypes, and idiotypes. Isotypes are due to differences in constant region or heavy chain and they are species specific. The allotypes are due to the allelic differences in the gene resulting variation in the amino acids in the constant domains of the heavy and light chains. The idiotypes may arise due to differences in the amino acids in the variable portions of heavy and the light chains. Amino acid difference creates idiotypes. 4 types of bonds: hydrogen bond, ionic bond, hydrophobic interactions, van der Waals forces, and ionic bonds. Affinity, avidity. Paratype, epitope. Affinity and avidity. Pentameric antibody showing avidity. The binding strength of a single epitope and paratope is called affinity, but the total strength of interaction of multiple epitopes and multivalent antibody is called avidity. Since the antibody can bind 2 to 10 epitopes of an antigen, the total strength is more than affinity of single antigen and antibody. So that is the difference between affinity and avidity. Some hotspots in the antibody research are designer AB, chimeric AB, antibody as immunosensors. Once again, antibody research at designer AB, chimeric AB, antibody as immunosensors. Protein sequence submitted, designing of antigen, synthesis of peptide antigen, production and purification of antigen, quality check by Western blotted ELISA, final product delivered. ELISA, immunoprecipitation, immunohistochemistry, immunostain, Western blot, etc. Showing production of designer antibody. Immunosensor provides signals using antigen and antibody. Sensitive, selective, inexpensive, USCD in clinical diagnosis, environmental analysis, food industry, uses and advantages. Immunosensor. Now let's have a quick summary. Antibody is the important effector molecule of the adaptive immune system. It has a Y-shaped glycoprotein with Fab and Fc components. It has diversity as it is specific to variety of antigens which are encountered in the blood and other body fluids. It is both membrane bound and secretory. Membrane bound antibody is for recognition of antigen and is part of recognition unit called BCR. Each component of AB, that is light and heavy chains, hinge region, disulfide bonds, J chain, and carbohydrate group gives it specific properties and makes it a versatile molecule. It has 5 isotopes as IgA, M, G, D, and E. Each type functions at strategic locations of the body. The various effective functions like ADCC, complement activation, opsonization, etc. aim to target the antigen and differ in isotype and subisotype. The AG-AB interactions are non-covalent and occur at the epitope of antigen and paratop of antibody. So that's all about this topic. I hope you guys understood. We'll be ending the session by once again thanking the developers of these notes. I express my sincere gratitude to them. Thank you so much. Thank you everyone for watching this video. Happy learning.